My name's Karl Maluf. I'm the director of the Biophysical Characterization Core. And of course, I'd like to start this talk out by thanking a very talented scientist in my group, Nikki Maklek. She's shown a lot of interest and done a lot of hard work on this project. All of the data that I'll show you and all of the data analyses that I'll show you were carried out by Nikki. And so big thanks to her. AAVs, adeno-associated viruses, are an increasingly common vector that are used in gene therapy process. By my last count, currently there are 6 of these that are approved by the FDA on the market, but there's hundreds of them that are actually in clinical trials. This is an important area for us to develop these sorts of capacities. These AAVs are composed of a proteinaceous capsid that encapsulates the single-stranded DNA genome. When these things are manufactured and purified, they typically are actually pretty heterogeneous. They contain differing amounts of various kinds of subspecies. Sometimes we see empty capsids. So this is the proteinaceous capsid shell without the single-strand DNA genome. We see various partially packaged capsids, and we've analyzed hundreds, maybe getting close to 1,000. I haven't actually counted that. We've analyzed a lot of these species, and we almost always see evidence for some sort of partially packed capsid. And this simply refers to a proteinaceous, capsid shell that has the full complement of the single-strand DNA genome packaged within it. So some shortened, perhaps clipped genome, perhaps actually non-specific DNA, whatever the case may be. Of course, we also see the fully packaged capsids. This, will represent the intended drug product. And of course, we also see varying amounts of things like viral and/or capsid aggregates, forming what we would call HMWS material. We also see capsid fragments forming what we would call LMWS material. These capsid fragments can correspond to things like misassembled or partially assembled capsids. Sometimes, depending on the sample that we're looking at, we actually will see a little bit of free-floating single-stranded DNA. And so, given this discussion there, it's clear that this heterogeneous nature of these purified AAVs, the characterization and quantification of these species is critical to ensure safety and efficacy of these treatments. We have to do a good job. In the field, there's currently 2 sort of workhorse techniques that we use to characterize these size distributions. There's some new exciting technologies that I haven't put on this slide here. There's mass spectrometry. We actually just purchased one of those. It's on our loading dock back in Louisville. But today I'm going to talk about AUC. I'm going to contrast that with AUC stands for Analytical Ultracentrifugation, and for AAVs in particular, it's an excellent technique to characterize the size distribution. The sedimentation velocity, that's SV, and in the AUC, the sedimentation velocity technique, in particular, will separate hydrodynamically and based on the size and the shapes of these species. When I say size, I mean molecular weight. Bigger things sediment faster when you spin them. Also, more compact things will sediment faster when you spin them. So sedimentation velocity will separate based on size and the overall shape of these things. Another technique that we use in the lab with the analytical ultracentrifuge is cesium chloride density gradient analysis. And so what you can do there is you put basically somewhere around 3 molal or so cesium chloride salt solution into the analysis cell. You put your sample in there, and then you spin that thing real fast, 60K, and that'll generate a steep density gradient across the analysis centerpiece. And since nucleic acids and proteins have differing densities, you'll actually get separation of various packaging states based on their density. So it's another way to separate. So on the one hand, we can separate hydrodynamically based on size and shape. Using the same instrument, we can also separate based on density of the species. Now, on the other hand, size exclusion chromatography, obviously coupled to a moles detection system, very important workhorse technique in the biopharmaceutical industry, as everyone knows. In the case of AAVs, it struggles to measure these size distributions. And the reason why, fundamentally, as we're all aware, size exclusion chromatography will separate things based on hydrodynamic radius of the species. And these various fill states have essentially the identical hydrodynamic radius. So you don't get any physical separation of these species on a sizing column. The methods that are out there to do things like calculate empty-full ratios are actually, all the equations that are out there that you use fundamentally assume that all you have in the main eluting peak is some mixture of empties or fulls. And as we just discussed, there's all different kinds of species that can be formed there, and so you're violating that assumption up front there. This work was inspired by a very important paper that came out a little while ago now, in 2021, by the UC lab. The lead author here is Maruno. And in this paper, they showed us how to use the new Beckman Optima analytical ultracentrifuge in order to calculate the relative amounts of protein and nucleic acid that are present in these AAV samples. And so this is possible with the new centrifuge because they fixed the problems that we had with the old models, the old XL model. Where with these old models, if you were trying to bounce around from one wavelength to another during a run, the precision of returning to exactly the same wavelength was not very good. It was on the order of a couple, plus or minus a couple of nano. And as a consequence of that, you had too much variation in the total absorbance values. When you analyze these things, you'd get a lot of artifacts as a consequence of that. While it was conceptually, theoretically possible to do these kinds of experiments for a while, practically it just wasn't something that made any sense to do. But Beckman fixed this problem. We now have very good precision for wavelength selection, and we also can scan along the radial position very, very rapidly. So this is now, it opens up the door to do these kinds of analyses and do it with some kind of reasonable throughput, at least for the AUC technique. So how does this thing work? I've got the picture of the instrument up here in the upper right, and then in the lower right, I'm showing a simple schematic of how the analytical ultracentrifuge is actually put together. And so it's basically an ultracentrifuge, as you would imagine, that's coupled to an absorbance detection system. As you can see, there's a light source. That light source shines against a diffraction grating, which will then select your wavelength. It's focused and it's bounced around on some mirrors, so that shines down onto the top of your analysis centerpiece. You've got light that's shining down on the top of the analysis centerpiece as the rotor is spinning, and then you can measure the relative amount of absorbance that is passing through the sample. In order to measure absorbance as a function of the radial position is along the radial axis of that rotor. There's a moving slit assembly. That thing moves around there, and then you can actually calculate your absorbance as a function of the radial position. With this moving slit. And so the kind of data that you might get from one of these experiments is shown here in the upper left. So on the y-axis of this plot, we have absorbance. Of course, that's a measure of the concentration. And on the x-axis, we have the radius in centimeters. And so this is the actual distance from the center of rotation of the rotor. For an experiment like this, you put your sample into your analysis centerpiece. The analysis schematic of that is shown on the bottom left. You put your sample into the analysis centerpiece, you spin it, and at the very beginning of the experiment, there hasn't been any sedimentation of sample, and so the concentration is basically the same everywhere along the analysis centerpiece. So as you're scanning across the radial position there, you're going to get the same absorbance value. But then as you allow these boundaries to evolve, you're going to see the formation of boundaries, which are then going to move from the left, from lower radial positions to higher radial positions as a function of time. And these boundaries that you're seeing up here are at increasing times. So it can scan very rapidly across the radial position relative to how fast these things are actually moving. And so each one of these boundaries essentially represents a snapshot. Now, what do we do with this stuff? The most common thing that we do, there's a lot of ways to analyze these kinds of data. The most common things that we do in the lab these days is we'll analyze it using the C of S analysis method. And this is incorporated into a software package called SedFit. It's a publicly available, freely available software package. It was written by Peter Schuck at the NIH back in the early 2000s. And what it does is it'll directly fit these sedimenting boundaries, and then it'll spit out a size distribution. So what you're seeing on the right here would be a size distribution that can then describe those sedimenting boundaries. And that happens through basic nonlinear least squares analysis of those primary data. So on the right here, we're showing, this is actually data on BSA. We're showing BSA monomer. There's some irreversible dimer in there, obviously the disulfide linkages, and some irreversible trimer. And so with this method, we can then quantify the relative amounts of monomer, dimer, and trimer that are present in this sample. On the y-axis of this plot, we have a quantity called the C of S. That's simply proportional to the concentration. You can think of these plots as probability density functions or histograms, if you will. On the x-axis, we have the sedimentation coefficient. The sedimentation coefficient is a physical property of a macromolecule that characterizes its size. Bigger things have bigger sedimentation coefficients, and it characterizes its overall gross structural properties. So things that are compact, like spheres, will sediment faster. Things that are elongated, highly extended, will tend to sediment slower. So the sedimentation coefficient is a measure of both the size and the shape of your species. To investigate how we were going to study these AAVs, using our new Beckman Optima. You see, I didn't mention we actually have 4 of these now. So we have 4 of the Optima AUCs in the lab. We've got 2 of the old XL models, and we actually have 1 of the really old XLA model from back in the 90s. Things still kicking. So we sourced some commercially available AAV. I'm not going to tell you the company because they sent us some stuff that was really heterogeneous. So you buy this stuff, it's supposed to be purified, but as you can see from the size distribution, there's a whole bunch of stuff in there. We sourced that commercially available at AV, and then we carried out a series of these sedimentation velocity experiments. And we analyzed the sedimentation here over a range of analysis wavelengths. So for these particular data here, we're showing wavelengths from 230 nanometers all the way up to 280 nanometers. And you see that on the far right of the plot there. These experiments were carried out under what's considered typical analysis conditions for AAV samples. We spun these at 16,000 RPM at 20 degrees Celsius. I'm pointing that out because, later on in the talk there, we'll talk about how we change those. The first thing that we see, of course, we've analyzed this over a range of wavelengths, and we see that the relative proportions of these peaks vary based on the wavelength that we're looking at. And this makes sense because, as I'm sure we all know, DNA, for example, has an absorbance maximum around 260 nanometers. Well, your protein will have a local maxima somewhere around 280 nanometers, and then if you go to something like 230 nanometers, you tend to get quite a bit more absorbance for things like protein samples. So the relative responses of the protein and the nucleic acid components are going to vary, quite dramatically with the analysis wavelength. We can take advantage of that in order to directly calculate the relative concentrations of the protein and the nucleic acid components as they're set. I'd like to kind of point your attention first to what I'm calling peak 4 here. This peak has a sedimentation coefficient a little greater than 100S. And what we know from the literature and from experience that if you actually had a wild type AAV serotype 5, and it had the full wild type genome in there, that's about 4.7 kilobases. We're going to get a sedimentation coefficient kind of in a normal aqueous buffer, something like PBS, of a little over 100S. Well, that's interesting that we're seeing that because the AAV that we purchased, actually, the intended genome, the intended packaged genome, is only 2.5 kilobases. So we expect that the molecular weight of that species is of course going to be less, and we expect to have a slower sedimentation coefficient, somewhere probably around where peak 3 is. Peak 3 is giving us sedimentation coefficient somewhere around 85S or so. We're also seeing what I'm labeling here as peak 2, that has a slower sedimentation coefficient, and then all the way down to peak 1, got a sedimentation coefficient of about a little over 60S. That's where we expect empty, or at least mostly empty capsids to sediment. So as we go across this, we're, a hypothesis, a very reasonable one, and generally how these things are interpreted is that at peak 1 likely corresponds to empties or something at least close to empty. There's a peak or shoulder that we're calling peak 2, likely corresponds to some sort of a partially packaged material. Peak 3 then, has more single-strand DNA packaged, and peak 4 would have even more single-stranded DNA. But the problem with this technique when you're only using a single detection wavelength is that there's actually multiple hypotheses that are possible at this point. This peak 4, which is sedimenting faster than we would have expected, this could be arising at this point, possibly due to overpackaging. So we've actually got more single-stranded DNA in the virus than the expected 2.5 kilobase genome. Or it can arise because we've got some kind of an aggregation event, some sort of association event that's happening with the smaller species. So for example, that peak 3 material might be associating with an empty capsid. And that's going to increase the molecular weight, which is going to cause it to sediment faster. And that's a formal possibility here. That peak 3 material could be interacting with misassembled capsids, for example, which could then cause it to sediment faster. There's a lot of possibilities like that. But we can't formally rule those possibilities out by collecting data at a single wavelength. We've got this wavelength data. What are we going to do with it? How are we going to analyze it? How are we actually going to quantify the relative amounts of protein and nucleic acid? And so again, if we focus our attention on, just start by thinking about what's peak 4, because we want to know the answer to that. So what is the molecular nature of peak 4? As we can see here, it's reasonably well resolved for peak 3. It's not baseline resolved, I will acknowledge that, but it's reasonably well resolved from peak 3. And so we can integrate across peak 4 to calculate the total absorbance associated with peak 4 at each of these 5 analysis wavelengths. And, I'm sure all of us are very familiar with doing those kinds of calculations. Okay, so to then analyze the relative amounts of protein and nucleic acid that's present in peak 4, we're going to start by just simply writing down Beer's law for a multi-component system. And so in equation 1, we're showing the total absorbance here on the left-hand side. And that is equal to the total absorbance that's coming from the total protein component. That's represented by C there for capsid concentration. Plus, the second term is the total absorbance that's coming from the DNA component. And so in equation 1, epsilon sub C is the extinction coefficient of the capsid. C sub T is the total capsid concentration, and L is the path length. For this particular case, the path length is 1.2 centimeters, so we know what that is. Similarly, epsilon sub D is the extinction coefficient of the DNA. D sub T is the total DNA concentration, and again, L is the path length. Now, one of the really nice things that they did in that Bruno paper is they took advantage of both the interference and the absorbance optical detection systems that are available on this instrument in order to calculate the extinction coefficients for a range of AAV serotypes. There's a lot of data in that paper that's very useful. So we actually have calculated extinction coefficients for empty capsids. We have calculated extinction coefficients for single-stranded DNA over a range of wavelengths. So we can take those extinction coefficients, stick them into equation 1 in order to do these kinds of calculations. So we know those things. Now, the insight here is to recognize that, okay, the relative extinction coefficients of the protein and the DNA are going to be strong functions of course, the analysis wavelength. But more importantly, the relative amounts of the relative values associated with these extinction coefficients is also going to vary with wavelength. So if we construct a ratio where we just divide one of these by the other, as a function of wavelength, we're going to see a strong dependence there. So that implies that we can linearize equation 1 in order to do some simple calculations here to actually calculate directly these total concentrations. So to do that, all we did is we just divided both sides of equation 1 through by the extinction coefficient of the DNA and the path length, and that gives us equation 2. As you can see, equation 2 is obviously in the form of a line. Plot the quantity on the left-hand side there, which is the total absorbance at each particular wavelength, divided by the extinction coefficient at that particular wavelength, divided by the path length. So that's the quantity on the left-hand side. And on the right-hand side, we're going to plot that quantity against the ratio, the extinction coefficient of the capsid and the DNA. And when we do that, we see that the slope of this plot is going to give us the total capsid concentration, again, for peak 4. And the intercept is going to give us the total DNA concentration for peak 4. So what we've done is we've calculated the total concentration of protein and the total concentration of nucleic acid for a peak. And when we do this, we like to present these results as the mass fraction of DNA. At the bottom here, that's simply calculated as you first expect, is the DNA concentration divided by the total concentration of the species. The total concentration of the species, for example, for peak 4 would just be the total DNA concentration and the total capsid concentration. So we're going to calculate the mass fraction of DNA as written there. All right, so when we carry this analysis out, it actually works really well. So when we're focused here on peak 4. So this linear data is shown for peak 4 over a range of analysis wavelengths. So as you can see on the plot, we've got 5 groupings of points here. Those are the 5 different analysis wavelengths. And then we carry this experiment out 10 times. We've got 10 replicates, and they're all overlaid. And what you can immediately see is that from the spread of the slopes, we're getting tight measurements of the total capsid concentration from those slopes. And from the intercepts, we're seeing really good reproducibility here for the intercepts. So we're getting a tight measurement of the total DNA concentration. And so from, of course, the slope of the intercept there, we can calculate the DNA mass fraction for this peak. And this is giving us a value of 24.4% plus or minus 1.2%. So as far as AUC, these kinds of analyses go, that's a real tight standard deviation there. Now, we care about this because, again, we know what the molecular weight of the AAV serotype 5 empty capsid is. And we know what the molecular weight of the 2.5 kilobase single-strand DNA is, so we can calculate the expected DNA mass fraction for a single genome packaged into the capsid. And when you do that calculation, that expected value is 17.2%. So what we immediately know now is that peak 4, the DNA mass fraction for peak 4, is significantly greater than what we would expect for a single genome packaged into the capsid. And so that's telling us that this, in fact, corresponds to overpackaged material. And in particular, it's telling us that this peak does not correspond to the alternative hypothesis that perhaps it's sedimenting faster because it's assembled with something like an empty capsid, for example, or perhaps additional misassembled capsid protein or whatever the case may be. Because if that was the case, we would expect that DNA mass fraction to be smaller, 17.2. which of course we didn't see. We can rule that hypothesis out, and all with a single measurement and a single instrument is simply taking advantage of the fact that we can measure these things now over a range of analysis wavelengths. So we can carry out the same calculation here for peak 3. Again, we acknowledge this is not baseline resolved, but it's pretty well resolved. So we went ahead and did the integrations there. And when we do that, construct a similar plot there, we calculate a DNA mass fraction for peak 3, 17.8% plus or minus 1%. And that is actually in really good agreement with what we'd expect for a single genome. So our interpretation of these data is that peak 3 corresponds to a single genome that's packaged into the capsid. Peak 4 corresponds to overpackaged material. Similarly, when you run this analysis for the smaller peaks there, of course, there's a lot more variability because there's not as much signal. But the DNA mass fraction for peak 2 is less than 17.2%. That's consistent with this being probably some distribution of partially packaged material. Peak 1 still actually comes up with a little bit of single-stranded DNA, but it's hard to know where the detection limits are at this point. Do those measurements. Okay, so this is cool, right? It's simple, the math's simple. All you got to do is run these experiments, fit each one of these wavelength data, to using SEDFit to get your C of S distribution. Then you can carry these calculations out. But we should be able to actually simultaneously analyze wavelength data at the same time, two size distributions that actually correspond to the total protein size distribution, as well as the total DNA size distribution. And if we look at this, we're just sort of thinking about, okay, I'm analyzing data here with nonlinear least square sorts of methods. Well, I've got 5 wavelengths here. That means I have to calculate 5 size distributions. And from those 5 size distributions, I'm going to calculate additional material. On the other hand, if I can analyze all 5 of these wavelengths at the same time, I'm only going to pull 2 size distributions out. So I've got more constraints, right? So it fundamentally is going to be a better approach here to globally analyze all these data together because we've got more constraints, we've got less degrees of freedom of nonlinear least squares fitting. That's a place that we want to get to for sure. And in order to do that, we used another software package that was put out by Peter Schuck. It's called SEDFAT. And as the name FAT implies, it's an extension of SEDFit that actually offers you a lot of different sorts of models that you can use in order to directly analyze your sedimentation velocity data. The model that's available in there that we care about is actually a multi-wavelength model. And so it was very hard. This is like a lot of freely available software. It's real powerful, but it's really hard to figure out how to use the thing. But we worked hard at that and figured out how to do it. And that sentence right there is a couple of months of banging our heads against this, trying to sort this thing out. But anyway, one of the limitations that we have with SEDFAT is that it actually only allows us to bring in 3 wavelengths currently. I'm not sure if Peter is planning on expanding this model, a significantly better software package here in terms of analyzing multi-wavelength data there. And that's actually something that we're in the process of bringing in-house. This is going to be for our GMP services, and so bringing something like that in, it's taking a long time. All the IT people have to get all that stuff sorted out. Anyway, that's coming in-house here. But currently what we can do is we can analyze 3 of these wavelengths at once and simultaneously in order to generate these 2 size distributions. And when we do that, it works really well, right? So as you can see, the sedimentation velocity data are well described by the fitted lines. It gives us good residuals as far as AUC data go. These are excellent residuals. And we're showing the data here for 230 nanometers, 260 nanometers, and 280 nanometers. And when we do that, we can actually pull out the total protein size distribution, that's the curve in red, and the total DNA size distribution, so that's the curve in green. And very interestingly, of course, the overall shape is similar to what we saw when we just analyzed the single wavelength data together. But very intriguingly, what we're seeing is better resolution for the total DNA size distribution, and that's that trace in green. And when we think about how single-strand DNA is going to behave in solution, this 2.5 kilobase genome, if it was actually not encapsulated, it's going to have a sedimentation coefficient of something like 20x or so far to the left, and nowhere near where these things are sedimenting. So it has to be encapsulated or otherwise associated with large capsid-like material in order to sediment this fast. So we're seeing it co-sediment with these protein species. And this also then immediately implies that rather than having to do this linear transformation analysis, we can simply calculate the areas under these peaks in order to directly measure the total DNA and the total protein concentrations, and make the same calculations for these peaks. Which this is one of the replicates for the 10 that I showed you previously, and these are the DNA mass fractions that we calculate from that, which are consistent with the numbers that we got from the linear analysis. So when we do this, now we did this analysis for all 10 of these replicates, and we're showing overlay plots here. Where in the blue, we're seeing the total protein concentration distributions, and in the red, we're seeing the total DNA concentration distributions. And we're seeing that reproducibility of these size distributions is good. As far as these sorts of data go, it's actually surprisingly good. In particular, the DNA size distribution is particularly reproducible. And so, a couple of things to note from this. In fact, it's probably a little easier to see it here. When we look at this peak right here for the DNA size distribution, lines up real well with this peak for the total protein distribution. This implies to us that this is likely a homogeneous species. Doesn't prove it, but it implies it. On the other hand, this peak right here does not line up as well. This DNA peak does not line up as well for the protein peak. And so this is hinting that there may be some heterogeneity actually present in this overpackaged material. Similarly, the shoulder is not well resolved here, and it's spread out, which is implying that we probably have quite a bit of heterogeneity here in this partially packaged material. When we go across these size distributions and integrate them and calculate these DNA mass fractions for all 10 of the replicates here, we're actually seeing once again that the reproducibility of our DNA mass fraction calculations is very good. So for peak 3 here, we're calculating a DNA mass fraction of 18%, plus or minus 1%, which agrees very well with what we saw with the linear transformation method, which was 17.8%. And it also then agrees with this peak 3, again, corresponding to a single genome package of the capsid. And similarly for peak 4, across all these 10 replicates, when we calculate the DNA mass fraction, we're getting 21.6% plus or minus 2%, which also is in good agreement with what we got from the linear transformation method. The next thing that we wanted to try is some experiments that were put out in a very recent publication. This is actually still a preprint here. And the authors of this publication call this high-speed sedimentation velocity AUC analysis for the purposes of analyzing these AUV samples. And the idea here is, well, these are very heterogeneous samples often, and can we do a better job of further resolving these fill states? And so in order to test this out, we actually wanted to try this high-speed experiment, but we wanted to try it using the multi-wavelength approach. In this preprint here, that they just had a single analysis wavelength. So we tried this high-speed sedimentation velocity analysis approach here over a range of wavelengths. We spun this much faster. So rather than 16K, we spun this at 45K. And we also lowered the temperature to 10 degrees. Now we spun it faster because the faster you spin these things, the better the separation you're going to get between these species. And the fundamental reason why is as these boundaries sediment, they're also diffusing. And the more diffusion you have in these boundaries, the harder it is to actually resolve species. This is similar to how you'd think about a sizing column. If you run a sizing column slow, it has the bands have more time to broaden as they're actually running through the column. A very similar principle here. So if you run these things slower, those boundaries can spread much more relative to how fast they're actually translating. So in order to improve resolution, we want to try to spin these things as fast as we can. But we can't spin them too fast, because if you spin it too fast, you can't collect enough data. This is a balance between not spinning it so fast that you can't collect enough data, and you can't actually detect things that are maybe really, really, really large that are going to just tell it instantly if you spin it too fast. On the other hand, you don't want to spin it too slow, because these boundaries are going to broaden. You're going to have a hard time resolving species. So anyway, 45K is what they came up with, so that's what we tried. We lowered the temperature. This actually has to do with the optima AUC. These sedimenting boundaries can actually mix sometimes stochastically, kind of like a turbulent flow problem. And it turns out 10 degrees, you're lowering the probability of these conditions here. Now, if you look at the data on the left, what we're seeing is, again, similar size distribution. We obviously have that peak 4, a little faster than 100S. And we've got, the peak that's sedimenting a little bit slower than 100S. And we've actually resolved, quite a bit of additional peaks here, perhaps corresponding to various amounts of partially packaged material. Interestingly, though, this is, when you're looking at just a single wavelength, when you actually do the global fit of these data in order to generate your total protein and your total DNA size distributions, we see that DNA peak splits. So very intriguingly, we're seeing this material here, from the point of view of the single-strand DNA, it splits into two peaks. This right here corresponds to what it looks like is, this peak right here, which we're calling peak 5 now. This peak right here corresponds to the single genome packaged into the capsid. And we've got two overpackaged species now that we're actually detecting here with this high-speed approach. Something that you're not going to see if you just do a single analysis wavelength, even at the faster speed. And in particular, something you're not going to see if you're spending, 16K. We can then calculate the DNA mass fractions across this peak using the total DNA size distribution as a guide. And as we do this, we see that the DNA mass fraction, as expected, consistently climbs up. Peak 5 here is the one that's closest to a single genome package, so the intended drug product. Peaks 6 and 7 correspond to two different species of overpackage. Maybe there's more, and we're only able to split it into two peaks here. But it is telling us that whatever this overpackaged material is, it's heterogeneous. There's multiple overpackaged viruses. This is just a zoom here to demonstrate the difficulty that we have with some of the peak assignments. The final experiment we did is we wanted to try to get a little bit of a better understanding of what this heterogeneity might actually be. So we carried out a cesium chloride density gradient experiment on this same material. Everything I showed you so far, everything was separated hydrodynamically based on size and shape. Now we're going to try to separate these things based on the density of these particles. Had a high cesium chloride salt solution that we squirted the sample into, spun it at 60K, established a real steep cesium chloride density gradient across the cell, waited long enough until everything stopped changing, so we're at equilibrium, and did our measurements. We collected a bunch of wavelengths, deconvoluted those wavelengths into our total protein and total DNA distributions. When you look at this, right around here at 1.31, at a density of 1.31, this is actually the density of the cesium chloride. It's hard to calculate. If you want to know how to do that, ask me. It's outside of the scope of this talk. So when you look right here, 1.31, this is where we expect protein to actually sediment in one of these instant gradients. This right here likely corresponds to our empty capsids. But as we increase in density, we're seeing a lot of DNA material, right? That's just creeping up here. So it looks like we have quite a bit of partially packaged material. So this is more like kind of a continuous distribution, looks to me, of the partially packaged material. Now, as we get to this main peak here, this presumably corresponds to the intended drug product. And as we continue up in density here, we're seeing the overpackaged material. So this confirms, the heterogeneous nature of this material and some of our conclusions. But the problem with the cesium chloride approach is that, we have to put these samples into a really high salt solution, and it's not clear the extent to which that salt might disrupt the size distribution. And so we always have to interpret these data carefully. But in general, these data are in agreement with the sedimentation velocity data that I just showed you. Main takeaways from my talk, people like to say that AUC is the gold standard for AAV analysis, and we definitely agree with that. There's some new exciting technologies out there that are going to be important to bring in-house, and we're in the process of doing that. But there still is not another technique out there that allows you to analyze these things under formulation conditions without modification. And that is AUC at the moment. So it's still the gold standard. But in particular, the gold standard is using the new Beckman-Coulter Optima AUC for characterizing these AAV samples, because you can spam it with a bunch of wavelengths and learn a lot more about your sample. This commercially sourced AAV was very heterogeneous, even though they told us it was purified. This high-speed sedimentation velocity analysis, well, it's going to have a very low throughput. If you couple it with the multiple wavelength analysis, you're going to probably be able to do a better job resolving species. And the multi-wavelength analysis in particular gave us additional information that was not obviously apparent for the data that was collected at a single wavelength. So we think not only is this an important addition in our analysis toolbox there. We think that we want to be running these things over a range of wavelengths so that we can potentially further resolve in particular what's happening at the level of the whole DNA distribution. And finally, you got to be a little humble here when you're interpreting these things. This peak interpretation is obviously complicated, and it can be further complicated by the fact that you can have co-sedimentation of material that are actually different species. Different species can have the same sedimentation coefficient. So it's possible that some of these peaks here, you're maybe overcounting something like the protein because there's protein components that are aggregated and happen to have a sedimentation coefficient similar to what you're getting for one of your packaged species. So that's all I got. Thank you for your time and attention, and I'm happy to take any questions.